Hi, my name is Abbas and I'm going to talk about the identification of the most influential nodes involving all topological dimensions of a network. I'll first very briefly describe the network and centrality measures. As you can see in the photo on the left hand side of the screen, a network is composed of nodes or vertices and edges. The gathering of several nodes within a network could result in the generation of some modules or clusters, as you can see in this figure. But one of the critical analyses in network science is the identification of key players within each module and the entire network. To this purpose, in graph theory and network analysis, we apply centrality measures and indices to identify the most important vertices within a graph. There are actually numerous centrality measures, but to name a very few basic ones, we can mention degree, betweenness centrality, and neighborhood connectivity. Also, we should consider that each centrality measure captures a specific topological dimensions of the network. For example, in the case of these centrality measures, degree is a local centrality measure, betweenness centrality is a global one, and neighborhood connectivity could be considered as a semi-local centrality measure. According to the previous literature and also our studies, some centrality measures suffer from inherent biases. Here we showed that betweenness centrality and collective influence, as two global centrality measures, have a positional bias within the network which is in contrary to the behavior of neighborhood connectivity. Thus, we selected the neighborhood connectivity as a good candidate for removing the positional bias of these global centrality measures. Also, to develop an influential node identification algorithm that captures all topological dimensions of a network, we adopted this pipeline to very precisely evaluate the association and dependence between 6 of the most important and commonly used centrality measures. These 6 important centrality measures include the degree, local edge index, neighborhood connectivity, cluster rank, betweenness centrality, and collective influence. And we actually tested the association between each pair of these centrality measures across 200 real-world and simulated networks. The analysis demonstrated that while some centrality measures are positively correlated with each other, several ones have negative correlations. Also, several centrality measures were significantly dependent on each other. This slide illustrates the pairwise association of the studied centrality measures across 200 networks. Amongst all, we focused on these 7 pairs of correlations. Based on these results, the 6 centrality measures were integrated using the addition and multiplication functions. Accordingly, spreading score emerged as the integration of 4 centrality measures. This score is actually an index that measures the potential of each node in spreading the information within a network. Similarly, hub score emerged as the combination of 2 centrality measures. And lastly, IVI, or the integrated value of influence, was produced based on the integration of spreading and hopness scores. In this work, we defined that the nodes with simultaneously high spreading and hopness scores are the most important influencers within a network. Then we assessed the performance of IVI in the identification of the most influential nodes in comparison with 12 other contemporary methods using the Serer model. To define the Serer model, we can say that Serer, or Seer-based influence ranking, is the combination of leave-one-out cross-validation technique with the conventional Seer model for ranking the true influence of network nodes. According to the receiver operating characteristic analysis, IVI outperformed all other methods in the identification of the most influential nodes in 10 different biological and randomly simulated networks. To summarize, we can say that IVI algorithm integrates 6 of the most important centrality measures in such a way that their strengths are synergized and positional biases are removed. Also, IVI algorithm outperforms other algorithms in identifying the most influential nodes, and accordingly, IVI could be a beneficial tool for all future network analysis, including systems biological studies. The IVI paper is published in Patterns, Cell Press, and Gold Standard Data Science Journal. Also, the IVI algorithm as well as the IVI-based network visualization function are available in the R package Influential, which is accessible on its GitHub as well as the CRAN repository. On top of that, the R package Influential has a comprehensive vignette including the tutorial videos. Additionally, IBI and its visualization functions are available as a user-friendly Shiny app on the Influential Software Package web portal. At last, I would like to highly thank my supervisors and those who helped me in this project.